It's our pleasure to have Dr. Aaron Cox here with us. His talk is going to be new leptin signaling mechanisms regulating islet function, implications for type 1 diabetes, coming to us from UT Health Houston and McGovern Medical School in Texas. And he's also located at the Center for Metabolic and Degenerative Disease. A little bio about him. He's originally from Canada. He obtained his PhD from the University of Western Ontario, studying the impacts of maternal nutrition on fetal beta cell development and regeneration. He moved to Houston, Texas, and Baylor College of Medicine for his postdoctoral studies, focused on beta cell regeneration for people living with type 1 diabetes, extensively exploring various drugs, GLP-1 analogs, putative hormones like beta-trophin, and physiological models like obesity. And subsequently, he was promoted to instructor while working with Dr. Sean Hardig, performing translational studies aimed at blocking the negative effects of inflammation on glucose metabolism and insulin sensitivity in type 2 diabetes. Last fall, he was recruited to University of Texas Health Science Center at Houston, where he's actively building his research group to study the influences of nutrient and hormone signaling on islet function, beta cell metabolism, and regeneration. His new paper, which I'll drop in the chat if anyone wants to look at, is discusses Aronafin, an RA drug that lowers leptin and improves insulin sensitivity. And he's a recipient of three very sort of major grants recently, an NADDK R01, leptin regulation of delta cell function, June 2023 to present, a DoD Discovery Award, novel leptin islet signaling pathway regulates beta cell function and proliferation, February 2023 to present, and an NIH Hearn Gateway Investigator Award recipient for 2023. GABP is a novel regulator of beta cell metabolism and proliferation. So it's well funded and setting up his lab. And we're really excited to hear about the work, and take it away.
Thank you for that wonderful introduction. I'm really excited to be here today and share a little bit more about our research and in particular, one area of interest related to type 1 diabetes. And I think the evolution of diabetes and pathology over the past many years is shaped as the treatments have been improving, in particular intensive insulin therapy. And I'll get a little bit more into that in a minute, but what I wanted to start off with is just sort of center us on this famous Eisenbarth model, showing the events, sort of the progression of type 1 diabetes. And we all understand and accept very well that there is a large genetic predisposition to type 1 diabetes, but the time to actually developing diabetes is quite variable, and it's centered on this idea that there's some triggering event, and we don't know specifically what that is. It's believed to be one of many factors that could include a virus or some environmental factor, toxin, among many other options, and there's a lot of intensive research in this area. But after this triggering event, there's initiation of this autoimmune process that leads to the infiltration of the islet by immune cells, and includes presence of autoantibodies in the serum. And over time, there's progressive loss of beta cell mass, which you can see by this decreasing slope in the light blue shade here. And it's not until quite later on, when you lose a very significant portion of the beta cells, that you present with hyperglycemia and other symptoms. And at this time, we already experience a major loss of beta cells. And so the goal is really to either replace the beta cells or find ways to make the beta cells work better and protect them from the immune system. So one really shocking thing is the prevalence of type 1 diabetes is increasing dramatically. According to the CDC, there is a 30% increase in type 1 diabetes just between 2017 and 2020. Think about that. That's only a span of 3 or 4 years, and it's increased by 30%. That's not really something that's occurring due to a genetic factor. The timeline for that is not compatible. And even when you look at children and teenagers, there's a 21% increase over a time period about 9 years or 10 years from 2001 to 2009. And this is a pretty vulnerable population. And overall, globally, type 1 diabetes is predicted to double by 2040. So there's a very tremendous need for new approaches and understanding of type 1 diabetes so we can effectively treat it. The big
Aaron, is that first bullet point, do you think that captures the COVID insult to the population?
I did wonder that, and I know there was a lot of studies going on whether or not the COVID virus gets into the beta cells or into the islet or not. And um, I think there's probably a lot more research and data points that need to be collected as we get farther out from 2020.
Yeah, well, a paper's coming out we're featuring a paper this Friday in our Week in Review newsletter at the very top of the list there. So if people are interested,
Very
it
cool.
looks to be a good one.
Awesome. So, what are the sort of driving factors behind the increased prevalence? And I talked about the triggering events, and one that I'm kind of focused on is environmental factors such as nutrition and diet. And obesity is certainly a global epidemic um, issue. And specifically in the U.S., you can see by this map showing that several states, most of the states, feature at least 30% of constituents having being classified as obese. And overall in the US, it's about 2/3 of the population that are dealing with being overweight or obese. And so this is obviously a great concern because obesity has obvious metabolic complications. And considering type 1 diabetes is already a disease in which glucose metabolism is impaired, the combination of the two sets up a pretty vicious cycle potentially. In just extrapolating to children and teens, we also see very significant prevalence of obesity in this population. From 1975 to 2020, you can see that the rates of obesity in children alone have increased from 4 to 20%. Now, when you put these two together and look at the prevalence of obesity in type 1 diabetes, there are a lot of studies, and I'm just highlighting a few. You can pick your favorite study. There are obviously social economic determinants that factor in, whether you're looking at a European population, which age group, and so forth. But there is some correlative evidence to suggest that obesity is similar or higher in individuals with type 1 diabetes compared to the general population. In this study from the Pittsburgh Epidemiology of Diabetes Complications is really interesting because they followed patients for 18 years and they saw an increase in prevalence of overweight from 29 to 42%, and an increase of 3% to 23% for obesity. So this really sort of begs the question of, how is type 1 diabetes influencing weight gain? And one of the sort of primary ideas is that the intensive insulin therapy is contributing to additional fat storage and better regulation of metabolism. There are a number of other factors, such as changes in hormones. Again, this kind of comes back to our younger individuals, teenagers that are exhibiting a lot of changes in hormones. One other hypothesis, and I'm not sure how much data there is on this thus far to really strengthen this idea, but when you're trying to battle hypoglycemia, you're always reaching for those sugary snacks, things that are very dense in sugar and may cause additional insulin stimulated fat storage. And then there are other factors, age, duration of diabetes, and potentially some genetics involved here as well. So there's this idea that type 1 diabetes may be driving obesity, potentially through insulin therapy, but it's also worth questioning whether obesity could potentially drive type 1 diabetes. And at least some groups have proposed this accelerator hypothesis, where If you are overweight or obese, or potentially with a family history of obesity or type 2 diabetes, you may be predisposed to type 1 diabetes. And then the mechanisms around this, are not quite clear yet, and I think more data is needed. But I'll just point out a few relevant factors here. Obesity is associated with chronic low-grade inflammation, increased cytokines, increased T cells and macrophages that are polarized towards inflammation. Obesity also induces insulin resistance and increased leptin levels. And all of these factors potentially could initiate, I won't say that for certain, but there is, more immune cells that are potentially in an environment that could activate them. Insulin resistance puts a lot of stress on the beta cell, and work from Dr. Emily Sims and Dr. Carmela Evans Molina have shown that in early development of diabetes, and type 1 specifically, there are definitely indicators of increased beta cell stress. And then I'm sort of looking a little bit towards leptin, and hyperleptinemia and obesity can cause a lot of different pathologies or impairments in energy homeostasis, and could potentially exacerbate the pathology or the progression of type 1 diabetes. So higher leptin levels is known to activate T cells, and we know also what I'll focus on today is that leptin can influence the beta cell. So we really need a better understanding of these factors in different settings, whether it's initiating or triggering type 1 diabetes, or just more importantly, the presence of obesity and type 1 diabetes. How is that going to affect progression? How does that affect our treatment approaches? So leptin is a satiety hormone. It's produced by the fat cells in our adipose depots, and it primarily signals to the hypothalamus in the brain to signal satiety or fullness. It helps increase energy expenditure, adiposity, and it's very important for reproduction as well. So it's part of this, balance between food intake and energy metabolism to sort of maintain weight. But what's really interesting to me is that leptin can inhibit the beta cell, and it's part of this sort of feedback loop where insulin is important for fat storage in the fat cells, and then leptin regulates insulin secretion. And so those data have been around for over 25 years. Tim Kiefer's group and Rohit Kulkarni show his data here in particular, showed very nicely that when you look at insulin secretion here on the y-axis, with changing glucose concentrations, you infuse leptin to islets and you can measure how much insulin is secreted, and there's a significant drop with leptin. And this is similar in human islets. You can see a dose-dependent effect of leptin on human islet insulin secretion. And as I mentioned, Tim Kiefer's data supports this as well. So this is pretty well accepted. But what we don't truly understand is the entirety of the effects of leptin on islet function as a whole. What is the mechanism, and how is it influencing the beta cell in particular? And then sort of more long-term, what is the impact of leptin and obesity in type 1 diabetes in terms of the islet? So the islet is this very intricate network of different cell types that communicate by paracrine action, and they form these different signaling loops. So our insulin-producing beta cells secrete a hormone, protein peptide called urocortin 3 that acts on the somatostatin-producing delta cell, which feeds back to inhibit insulin secretion. Similarly, with the alpha cell, glucagon can activate the delta cell to secrete somatostatin to inhibit its activity. And then glucagon can actually potentiate insulin secretion, which feeds back to inhibit the alpha cell. So what's really important is understanding how this network is regulated. How do external factors and nutrients and hormones influence this? And we know that the leptin receptor in the rodent islet is expressed on all three major cell types. And so leptin can suppress insulin secretion. It also suppresses glucagon. But are these direct actions, and what is happening in the delta cell? Because we know the leptin receptor is expressed there as well. And this is important in the setting of obesity because leptin is chronically elevated. So potentially we're exacerbating this suppressive effect on the alpha and the beta cell. Not too long ago, Guy Rutter's group published this paper showing that if you knock out the leptin receptor on the beta cell itself, you see no impact on islet function. So this is glucose stimulation. In the control and the knockout, they have similar secretion despite the lack of a leptin receptor, and there's no impact on glucose homeostasis. So the first thing we had to do was sort of validate whether, leptin actually could suppress insulin secretion, and the answer is yes. So all of those early data are still valid, and this is a perifusion experiment where you add in leptin, and as the glucose concentration increases, just we saw before, there's a suppressive effect of leptin to lower insulin secretion. So that part is very well and good, but what is the underlying mechanism? How is leptin acting to inhibit insulin secretion? Well, we know from the single cell data from human islets that the leptin receptor is actually enriched on the delta cell. Here you see this green spike here on this violin plot. And it's very low or absent in the other cell types. And this study from Lawler et al., they showed by our in situ hybridization that the leptin receptor, green here, does co-localize with somatostatin. So this led to our hypothesis that leptin is actually acting through the delta cell to regulate alpha and beta cell function through somatostatin. And this has become a pretty well-described paracrine action for ghrelin and fatty acids and other things. So the delta cell really appears to be sort of this overarching governing cell of the islet. And so it makes sense that leptin and other hormones might influence the same pathway. So here are some static incubations with leptin using human islets and measuring somatostatin secretion. So we're showing here that compared to vehicle, that leptin can actually stimulate somatostatin secretion. And this correlates with a reduction in insulin secretion. And we can do this also by perifusion. So you can look at the leptin group in red here, and you see that soon after the administration of leptin, somatostatin levels go up. And this temporally correlates very nicely with a drop in glucagon. So we went on to make a delta cell specific leptin receptor knockout mouse. So we use a somatostatin promoter to drive Cre expression to knock out the leptin receptor through a constitutive Cre recombinase activity. So in wild-type mouse islets, much like I showed for the human islet data, you can administer leptin in these static incubations and see an increase in somatostatin secretion. However, when you knock out the leptin receptor, this effect is largely lost. So in the absence of the delta cell expressing the leptin receptor, we lose that stimulatory effect on somatostatin. So in contrast to some of the beta cell work, we're seeing some direct effects here. Okay, so that work is ongoing, and we have a wonderful new postdoc in the lab by the name of Tanya Ashik, who's going to be carrying out some additional work on that area. But we're also looking to understand the mechanism and what's downstream of the leptin receptor that might be influencing somatostatin secretion in the delta cell in general, because we don't have enough understanding yet compared to alpha cells and beta cells as to what regulates delta cells. And so in the control here, these are some single cell data using control and whole body leptin receptor knockouts. So in the absence of leptin receptor expression on different cell types, we can look at the gene profiles, and here we're focusing on the delta cell. And these are the top enriched transcription factors. And through unbiased means, we pulled out a very well-known leptin receptor target, STAT3. So STAT3 is part of this canonical JAT-STAT signaling. And when you look at data of purified alpha, beta, and delta cell populations, STAT3 is the highest expressed in the islet. So we figured we were on a good track here, and we decided to take some human islets and treat them with leptin, and just look at a couple of obvious targets downstream of STAT3, which includes STAT3 itself, as well as the ATF3 gene. And you can see in the presence of leptin, we see increased expression of these two targets. And then we're also looking at activation of STAT3 through staining for phosphorylated STAT3. So here's just a couple representative images. You can see in the vehicle versus the leptin, there's nice co-localization of phosphorylated STAT3 in the nucleus. So continuing this work in mouse studies, we've similarly generated a STAT3 knockout in the mice and performed some static glucose incubations and looked at somatostatin secretion. Again, in the wild type islets, we can induce somatostatin secretion. But when you knock out the STAT3 in the delta cell, that effect is blunted or nearly absent in most cases. So just to understand this, because STAT3 is a transcription factor, and its canonical activity is through regulating transcription, we just want to see maybe there's some underlying effects on islet architecture, because this is a constitutive knockout. So the gene is knocked out for all of life. And we didn't really see any changes in the delta cells that are shown here in purple or in the white here on the left. When we looked at the organization, they were pretty similar as well. So just to summarize where we are so far, we've shown leptin induces somatostatin secretion from both mouse and human islets, and this has been validated in other labs now. And we believe that this really unifies a mechanism whereby leptin suppresses insulin and glucagon secretion through stimulation of delta cell somatostatin secretion. So we're finally connecting some of the dots dating back to 27 years ago in those initial observations that leptin suppresses insulin. And then these have physiologic implications, because leptin always increases after a meal, and so there's potentially nutrient or organismal metabolic status being conveyed to the islet. And then this is significant as well in terms of type 1 diabetes, where we're seeing higher rates of obesity, and elevated leptin may further compromise beta cell activity in a setting where beta cells are already few in number and working very hard to manage glucose levels. And they showed that STAT3 is also important for delta cell activity, and more to follow on that. So I'll end there with some acknowledgements to my wonderful new group, including Tanya, El-Sanem, and Mariana, and formerly a lot of my mentors and colleagues at Baylor College of Medicine in Sean Hardig's group, and wonderful support and collaboration from Jonathan Campbell's group at Duke, and Patrick McDonald and Mark Husing at University of Alberta and UC Davis, respectively. Definitely have to thank the islet donors and their families, and support from the Alberta Diabetes Institute Islet Corps, as well as our funding sources. And I'd be happy to take any questions.
Yeah, no, that's wonderful. Thank you for bringing everyone up to speed on what you've got cooking down there in Houston. That's really exciting.
Thank you.
And just to sort of throw this out there, are you still hiring, looking for graduate students, postdocs, trainees, and techs?
Yeah, of course. I have one more exceptional PhD student coming as a postdoc in the summer. I think we'll probably hold tight in the short term, but we're always looking for talented young scientists to join our group, and certainly as we pursue new funding opportunities, there will be positions in the near future.
Great. And so, yeah, feel free to reach out for collaborative purposes or just to see, what opportunities might be arising in the future. I don't want to throw a fly in the ointment here, but when I was sort of reading the background literature on, to support your talk, I did come across this paper coming out of um, Penn and Hopkins, and I just drafted in the chat, but basically it's the leptin receptor has no role in delta cell control of beta cell function in the mouse. That's Maria Golson et al. Uh, and I wondered what you thought about that, because that's sort of like a different result in a way.
Yeah, for sure. And Maria and I have chatted, and she's presented some nice data at the American Diabetes Association last year. And I think there are always nuances to mouse models and other factors that are involved. I feel like our data is pretty strong, especially when we knock out both the leptin receptor and its downstream signaling factor, STAT3, that there seems to be a strong implication for this pathway in the delta cell. And so I think further work is needed to validate this, because nobody can argue against the amounts of data that show leptin suppresses insulin secretion and beta cell activity, and the mechanism is there at hand. I think what we found so far leads us to continue forward, and I'll continue to talk to Maria and see if we can sort of resolve this in the long term.
Yeah, so maybe just tease out the nuances. There can always, sometimes there can be duality or in a path, and then it's overlooked. But yeah, that's really interesting, and I think it's a great story. I wondered, just a tiny, tiny bit on your exciting new grant that's targeted at GABP. We know GABP is an ETS transcription factor, right? It's required in a ton of different things like mitochondrial biogenesis and controls T cell homeostasis and all this other long list.
Right.
But, it's kind of interesting, like you're thinking about it in terms of the changes. It's like a rate limiting transcription factor that responds to metabolic demand, right? So um, what happens, has anybody looked at the GABP in context of pregnancy and islet changes?
So there's very limited data in the islet in general.
Mm-hmm.
There's a couple high-fat diet studies looking at obesity, and they picked up on this transcription factor in their omics studies.
Mm.
Uh, Nothing that I've seen in pregnancy, though.
Yeah, it's kind of interesting because that is when the islet goes under a lot of metabolic demand. And
Definitely.
yeah, I did wonder, I guess, I don't know if you want to talk a little bit about that, but we are going to, next week, we're talking with Federica Vecchio, and her group, a large group, put out the new paper regarding CVB and its impact on T1D, sort of further defining its role.
Okay.
And I wondered, has anyone looked at the CVB effect sort of on that whole, the triad of the alpha, beta, delta, leptin communication pathway? Is that anything of interest, or anyone's looking at it as far as you know?
Not to my knowledge. I do think that's a really interesting question, in something potentially to look into. Uh, there's always a challenge when you're going from mouse to human, right? And
Right.
the mouse studies, the delta cell population is so small. And I think while we root a lot of our work in mouse models, we're pushing more on human islet studies nowadays, and really trying to understand the role of the delta cell, because it is a little bit more prominent, and its cytoarchitectural positioning within the islet supports that as well. And so, uh, as I alluded to earlier on, the big question is, what are some of the factors regulating this intricate network? Some of them have been identified, such as ghrelin and free fatty acids, and Mark Housing and Mrs. Poitou have done a lot of great work in this area. But I
Yeah.
think we're just still scratching the surface in this area.
Yeah, no, it is. I'm there. It's really interesting. They have those delta cells, even their cytoarchitecture, the way they have that little probing, phenotype, kind of almost like checking on the other cells, testing them.
Well, and now there's a lot of work on cilia in the islet cells, and it's another appendage or another sensory component,
Hmm.
exploring that microenvironment. And so the better we understand the communication between cells, I think we'll be much further along.
Yeah, new paper recently coming out that sort of the polarity of the beta cell with the cilia at the one end.
Hmm.
And it's not there for nothing. I'm
Yeah.
certain. So anyways, yeah, okay, last call for questions. Anyone? All right. Forever. Hold your peace then. You can reach out directly to Dr. Aaron Cox at University of Texas, Houston, and we wish you the best. We cannot wait to see what comes out of your lab next. It looks like you have a great crew there, and as I referred to earlier, some great funding. So it's a big vote of confidence for your work. So thanks
Very
again
good.
for joining us, and we look forward to talking with you again soon.
My pleasure. Thank you so much.